Have you ever wondered why you look similar to your parents, yet not identical? This is the magic of genetics at play. It's a fascinating field of study that unravels the threads of our biological tapestry down to the very core of our existence. Let's take a quick stroll down this avenue of science, shall we? Genetics is essentially the study of heredity. It's how traits like your mother's eyes, your grandfather's nose, or even your great-aunt's love for classical music are passed down through generations. You see, every living organism carries a set of instructions, the genetic code, that determines their physical characteristics and even some aspects of personality. These instructions are stored within our genes, tiny units of heredity that reside in every single cell of our bodies. And each gene is nothing more than a specific sequence of molecules, called DNA or deoxyribonucleic acid if you want to get technical. Imagine DNA like a long, twisted ladder, where the rungs are made up of combinations of 4 different molecules: adenine, thymine, guanine, and cytosine. These molecules, or bases as they're commonly referred to, pair up in a specific way, adenine with thymine, guanine with cytosine, to create the unique genetic code that is you. It's this code, this sequence of base pairs, that determines whether your eyes are blue or brown, whether you're tall or short, whether you're prone to certain diseases, or even whether you're more likely to be an introvert or an extrovert. And while our genes do shape us, it's important to remember that they don't define us. They're just a part of the complex interplay between nature and nurture that makes us who we are. So our genes, packed within our DNA, serve as a blueprint for who we are. But what if we could change that blueprint? What if we could tweak our genes, edit our DNA, to alter our traits, cure diseases, or even enhance our abilities? Stay tuned as we delve into the world of genetic engineering and the revolutionary technology known as CRISPR. It's going to be a ride you won't want to miss. Imagine DNA as a twisted ladder. How does it function in our bodies? Well, to understand that, we need to take a closer look at the structure of this ladder. DNA, or deoxyribonucleic acid, is made up of 2 long strands. Each strand consists of a sugar-phosphate backbone with a series of bases sticking out. These bases are like the steps of our ladder, and they come in 4 types: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases pair up in a very specific way. Adenine always pairs with thymine, and guanine always pairs with cytosine. This is the fundamental rule of base pairing in DNA. Now, the sequence of these base pairs is incredibly important. Why? Because it's the language of life. It's like a book of instructions where each base pair is a letter, and the sequence of these letters forms words, sentences, and paragraphs that tell our cells what to do. For example, let's say a sequence of base pairs spells out the word insulin. This word instructs the cell to produce the protein insulin, which is crucial for regulating blood sugar levels. This is how our genetic information, stored within our DNA, is used to produce proteins, the building blocks of our bodies. And it's not just about producing proteins. Our DNA also determines how these proteins function and interact with each other. It dictates the color of our eyes, the texture of our hair, our height, and so much more. It's also responsible for our susceptibility to certain diseases. In essence, our DNA is a complex and intricate code that governs every aspect of our bodies. But here's a thought. What if we could edit this code? What if we could change the sequence of base pairs to alter the instructions given to our cells? Could we potentially cure genetic diseases or enhance our physical abilities? That's where the fascinating field of CRISPR technology comes into play. So our DNA is a complex code governing our bodies, but what if we could edit this code? Enter CRISPR, a revolutionary technology that allows us to do just that. As we delve deeper into the world of genetics, we come across this intriguing acronym, CRISPR. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. Quite a mouthful, isn't it? But don't let the long-winded name intimidate you. The concept behind it is truly fascinating. Picture this. You're a bacterium, the microscopic single-celled organism. You're going about your day when suddenly you're invaded by a virus. What do? If you're a bacterium with a CRISPR system, you've got a secret weapon. You can actually remember the virus that invaded you and build a defense mechanism against it. This is the basic premise of how CRISPR was discovered. Scientists, while studying bacteria, observed this intriguing phenomena. They noticed that certain sequences of DNA in the bacteria were repeating themselves in a peculiar way. These sequences were separated by spacers, pieces of DNA that matched the DNA of viruses that had previously attacked the bacteria. The bacteria were essentially keeping a record of past viral invaders, using this information to recognize and destroy the viruses if they ever dared to attack again. Upon this discovery, researchers began to wonder, could this bacterial defense mechanism be used in other ways? Could we perhaps harness the power of CRISPR to edit our own genetic code? Imagine the possibilities. We could potentially fix genetic errors responsible for diseases or enhance certain traits. The idea was tantalizing, and thus, the journey of exploring CRISPR as a genetic editing tool began. So, in essence, CRISPR is a natural defense mechanism found in bacteria that has been repurposed for genetic editing. It's like a pair of molecular scissors, capable of cutting and pasting DNA at specific locations, allowing us to modify the genetic code of living organisms. As it turns out, what bacteria have been using to combat viruses can be a powerful tool for us to edit our own genetic code. And with that, we've just skimmed the surface of the vast ocean of possibilities that CRISPR presents. So buckle up as we dive deeper into how this fascinating technology works in our next segment. So, how does CRISPR, a system discovered in bacteria, help us edit genes? Well, the key to understanding this lies in the CRISPR-associated protein 9, more commonly known as Cas9. Think of Cas9 as a pair of molecular scissors. These aren't your ordinary scissors, though. They are precise, able to cut at specific locations within the vast expanse of our DNA. To understand how Cas9 works, we first need to delve a little into the structure of DNA. Our DNA, the blueprint of life, is a long, winding double helix filled with 4 chemical bases: adenine, thymine, cytosine, and guanine. These bases pair up to form the rungs of the DNA ladder, with adenine always pairing with thymine and cytosine with guanine. Now here's where the magic of Cas9 comes into play. Scientists can program Cas9 with a piece of RNA, a molecule similar to DNA, that matches the sequence of the DNA they want to edit. This guide RNA, as it's called, leads Cas9 to the exact location in the DNA where an alteration is desired. It's like giving Cas9 a GPS coordinate within the sprawling city of our genome. Once Cas9 arrives at the desired location, it does what it does best. It cuts the DNA. But this isn't a random slice and dice. Cas9 makes a precise cut at a specific point in the DNA sequence, breaking the DNA ladder in half. But what happens after the cut? Well, our cells have a natural mechanism to repair such breaks. However, this repair process isn't perfect and can lead to changes in the DNA sequence at the cut site. These changes can disrupt the function of a gene, effectively turning it off. This can be useful if the goal is to disable a harmful gene. Alternatively, scientists can provide a piece of DNA with the desired sequence along with the Cas9 and guide RNA. When the cell repairs the cut, it can incorporate this new sequence into the DNA. This allows scientists to introduce specific changes or even add new genes. So to sum it up, CRISPR and Cas9 allow us to target specific sections of our DNA, make precise cuts, and introduce changes. This system, derived from the immune response of bacteria, has become a powerful tool in genetic engineering. It's like having a word processor for our genes. We can delete words, add new ones, or change existing ones. But instead of editing text, we're editing the very code of life. It's a revolutionary technology that has opened up vast new possibilities in genetics and beyond. By harnessing the power of CRISPR and Cas9, we can specifically target and alter sections of DNA. But what does this mean for us? Stay tuned as we delve deeper into the implications and future prospects of this groundbreaking technology in our next segment. The ability to edit our genetic code has profound implications. Imagine a world where genetic diseases are a thing of the past, where our crops are resilient against pests and weather extremes, where we can even design our offspring's traits. These are just some of the potential uses of CRISPR, the revolutionary tool that allows us to edit our DNA. First, let's talk about health. With CRISPR, we could potentially treat or even cure genetic diseases. Conditions like cystic fibrosis, Tay-Sachs, or Huntington's disease, which are caused by mutations in a single gene, could be fixed by replacing or repairing the faulty DNA. This could also extend to more complex conditions such as cancer or heart disease, where multiple genes might be involved. Imagine the impact this could have on global health. It's like having a word processor for our genetic code. In the realm of agriculture, CRISPR could be a game changer. By editing the genes of crops, we can make them more resistant to pests, diseases, and extreme weather conditions. This could lead to more abundant, reliable food sources, particularly in regions that are currently struggling with food security. Furthermore, we could potentially enhance the nutritional value of crops, making our food healthier. But CRISPR isn't just about fixing problems. There's also the potential to enhance, to improve. This is where we start to venture into the territory of designer babies. In theory, we could use CRISPR to choose our children's traits, from their eye color to their athletic ability, or even their intelligence. However, with these potential uses comes a host of ethical considerations. Do we have the right to play God with our genetic code? Is it ethical to create designer babies where only the rich can afford to enhance their offspring? And what about the potential risks? While CRISPR is a powerful tool, it's not perfect. Unintended mutations could occur with unforeseen consequences. Moreover, there are concerns about the long-term effects of genetic editing. We don't yet fully understand our complex genetic code, and making changes could have ripple effects that we can't predict. For instance, if we eliminate a disease gene, could that have other unexpected effects on the person's health or their offspring's health? And let's not forget the potential for misuse. In the wrong hands, CRISPR could be used for nefarious purposes, from creating bioweapons to eugenics. These are not easy questions, and there are no clear answers. But what is clear is that as we move forward with this technology, we need to have these conversations, to consider the ethical implications, and to put in place regulations to prevent misuse. With great power comes great responsibility. As we advance in our understanding and use of CRISPR, we must tread carefully. What does the future hold for genetics and CRISPR? As we venture into the future, we can expect the field of genetics to be revolutionized by the continued refinement and application of CRISPR technology. Ongoing research is already leading us into bold new territories. Scientists are developing more precise and efficient versions of CRISPR, enabling them to perform genetic edits with unprecedented accuracy. Imagine a world where genetic diseases like cystic fibrosis or Huntington's disease are things of the past. A world where we've literally cut these afflictions out of our genetic code. This is just the tip of the iceberg. Beyond medical applications, CRISPR could transform agriculture, allowing us to engineer crops that are more nutritious and resistant to climate change. But it's not all rosy. As we push the boundaries of what's possible, we also need to consider the ethical implications. Should we use CRISPR to enhance human capabilities? If we can, does that mean we should? These are questions we can't afford to ignore. It's crucial to maintain an ongoing dialogue about the ethical use of genetic editing. As we unlock the secrets of our DNA, we hold in our hands the power to shape life itself. This power comes with a profound responsibility. The potential of CRISPR is vast, but so are the challenges. It's a tool, and like any tool, it can be used for good or ill. As we continue to explore its potential, we must do so with caution, respect for life, and a deep sense of responsibility. There's a lot to be excited about as we look to the future of genetics and CRISPR. It's a future filled with promise and potential, but also fraught with ethical dilemmas. We stand on the brink of a new era in human history, one where we have the power to direct our own evolution. The future of genetics and CRISPR is as exciting as it is challenging. As we continue to explore and innovate, we are writing a new chapter in the story of life itself. To summarize, we've embarked on a fascinating journey into the world of genetics and CRISPR today. We've peered deep into the complex and intricate structure of our DNA, the very blueprint of life. We've delved into the fundamentals of genetics, understanding how traits are passed on from generation to generation. Then, we ventured into the world of CRISPR, a groundbreaking technology with the potential to rewrite the code of life itself. We've seen its immense possibilities, from curing genetic diseases to potentially creating custom organisms. But with such power comes great responsibility. We've also touched upon the profound ethical implications of genetic editing. It's a delicate balance, with the potential to either unlock unprecedented advancements or plunge us into unforeseen consequences. As we stand on the brink of a new era in genetics, remember, knowledge is power. Stay curious, stay informed, and continue to question the world around you.